My name is Jackie Stevens. I'm a professor in the biology department at LSU, but my research lab is over at Pennington Biomedical Research Center, where I'm standing today, and here I'm the endowed chair of biomedical research. So my research effort is split between LSU and Pennington Biomedical, and so I teach at LSU, but my graduate students from LSU actually work in the laboratory over here. So we have one of 3 NIH-funded botanical research centers in the country, and what we try to do in that center is to identify plants and components of plants that might be able to combat metabolic disease states like obesity and type 2 diabetes. And so I have researchers and graduate students and undergrads working on that project. And the project that I've been working on for the longest period of time has to do with basic fat cell biology. So pretty much everybody knows what fat cells are. They store lipid, but they're actually not quite that inert. Most people who are type 2 diabetic are obese or have excess fat, but most people who are obese don't have type 2 diabetes. And actually their fat tissue is pretty healthy. So we try to compare fat tissue between people who are metabolically healthy and people who aren't, or animals that are and then animals that aren't. So to try to figure out at the biochemical and molecular level what signaling pathways and hormones are contributing to the pathogenesis of type 2 diabetes, we study unhealthy fat and healthy fat. And we do this with a two-pronged approach. We do experiments in cell culture where we're just looking at an individual cell population, meaning we're just studying the adipocytes without all the complications of the nervous cells and the vascularization. But then when we figure out something where we can perturb it at the genetic level and it can cause or protect against type 2 diabetes, then we shift that project into the whole animal, and we make what we call a transgenic mouse, where we manipulate the embryo of a mouse at a stage where you change that gene, it's going to be reflective of altering that gene in the whole animal when it's an adult. So we've been able to manipulate genes so that we just alter their expression in adipocytes, but not in any other tissue, so not in the liver or the muscle or the brain. And even just altering one gene in the fat cell of an animal can have systemic effects that can either promote type 2 diabetes or protect against diabetes. And so I've been studying fat cell biology for over 25 years. A long time, and what we really try to figure out about fat cells is how, if we change a gene or the regulation of a gene, how that can contribute to the pathogenesis of type 2 diabetes. And I think that's a particularly pressing issue in Louisiana because unfortunately type 2 diabetes is an epidemic, and you know, in the 1950s, less than 1 million people, or less than 1% of people in the US had diabetes, and now it's over 8%, except in Louisiana, where in every parish in the state, the incidence of diabetes is over 10%. So we do really struggle with this disease more than the average US state. And so that's one of the reasons I particularly care about trying to figure out what's going on with this disease that can be very debilitating.